Hello my dear friends and my dear students, welcome back to top lesson for you. Dear friends, as we are discussing about the cell cycle, right? So we had started to discuss about the cell division, but before cell division we need to have discussed about the cell cycle. What is cell cycle? In the previous lecture I have in a big detail discussed about all those molecular changes which occur inside the cell and which leads to the replication of the DNA, so that the DNA could be transmitted to two cells in equal size and shape and everything, right? So this was our previous lecture. You can go and watch this lecture, that what happens when a cell decides to go to division, right? What happens when a cell is going to divide, or what happens when a cell is not going to divide. So we have discussed all those in a big detail. Here you can watch those lectures. Okay, now let us have a very brief concept about the cell cycle, that what is cell cycle, and this was the molecular events which were occurring inside the cell. In the DNA certain genes are activated, and what does those genes do? I have discussed them in a big detail. Now in a briefly, we should know that when a cell is going to divide, when a cell decides to go to division, what happens inside that cell, right? So let us see here. First of all, friends, this is a cycle and we call this cell cycle. For example, we call this cell cycle. So look carefully. Here a cell comes, right? Look carefully, my dear friends. For example, this is a cell and this cell has decided to go to division, right? This is the DNA of that cell, right? So what happens when this cell has decided to go to division and divide into two cells? What happens to this cell, right? Let's see. In the previous lecture, I had told you that whenever a cell decides to go to division, it jumps to interphase, right? So let us very briefly revise that. When a cell, right, wants to divide into two cells, that will be seen later. What is mitosis? I will discuss later. But first we should have a clear concept of interphase, right? So these two cells are formed from this cell, right? So first of all, this cell should enter to a preparatory phase, and that phase is called the interphase. You know clearly. If you don't know about this interphase, you can go and watch that lecture so that to have a clear concept. Look, if the cell decides to go to division, then it must enter to a phase which is called interphase, and this interphase is called the preparatory phase, right? So look, whenever the cell enters to interphase, so there are three phases of this interphase. How many phases? Three subphases. The first one we had told you that is G1, right? You will see here. Then the next one is called the S phase, right friends? And the third phase, which was very important, which were the phases of the interphase, was G2, right? After the G1, G2, S and G2 completes, then the cell enters to another phase, and that phase is called the multiplicative phase or mitosis or division phase, right? So we are going to discuss right now here this G1 phase, G2 phase, and this G1 phase, S phase, and G2 phase, that what happens here in the cell when it jumps to interphase. Clear, friends? Look, at molecular level what happens we have discussed already. Now we will see generally that what should happen to the cell whenever it decides to go to division. Clear, friends? Listen to me, friend. From here, right, let me use red color. From here, when the cell jumps to a division phase, from here the cell will start and will reach Look carefully, the cell has jumped and the cell comes up to here, right? So all this time which the cell spends here and prepares itself for division from here up to here, all this phase is called the interphase. Clear, friends? So I should write it as this is our interphase. Clear, friends? So this whole step which consists of 3 subphases, this is called our interphase. Now the interphase I have written here consists of how many subphases? It consists of 3 subphases. There is G1, right? So here I will mention what is this phase. This phase is our G1 phase, right? And this phase is the S phase, right? And this phase will be our G2 phase. Clear, friends? So the interphase consists of G1, S and G2 phase. Clear, friends? This G stands for gap, right? Gap 1 or gap 2, and this S stands for synthesis. Synthesis. What is synthesized here? We will see. Or this S is also known as post G1 phase. Post G1 because it occurs after G1, right? And this S is also known as pre-G2. Pre-G2 phase. Pre means before G2 phase. Don't go in that. Let's see here, friends. Look carefully. What happens here? First of all, let me make your concepts clear that what happens in S phase, so that we will see that whatever happens in the S phase, preparations for this S phase have been done here. Look, if you have decided to cook a biryani or a very sweet and beautiful dish for yourself or for your guests, what you do first of all, you decide that what should I cook now. Whenever you decide, then you make the list for what substances are required for what you have decided to cook, right? So look carefully. Here we are going to cook DNA, to make DNA, to make an extra copy of DNA. So to make DNA, you know that DNA is made up of nucleotides. So you have to prepare here nucleotides first, right? You need enzymes to replicate the DNA. You have to prepare those enzymes here, right? So whatever changes occur in G1 phase, I want to tell you, friends, that whatever changes occur in the G1 phase, all these changes are for S phase. Clear, friends? Whatever happens in the S phase is dependent on G1 phase. How? Look carefully. Here what happens? Whenever the cell jumps from G1 phase to S phase, this is a cell. Clear? And when it enters which phase? S phase, what happens to this cell? Look carefully. This is the nucleus and this is the same cell. Look, what happens during S phase? Look, this is the DNA of that cell. Clear? So whenever the S phase starts, look carefully what happened here. Clear? So we have replicated the DNA. So during S phase, what happens? Replication of DNA. Clear? Now listen carefully. Whenever you want to replicate the DNA, so you have to make the copy of the DNA. So whenever you want to make the copy of the DNA, whatever nucleotides are present in this DNA, those nucleotides should also be present in here in this DNA, in the next forming DNA. So from where does these nucleotides come? These nucleotides are synthesized in G1 phase. Clear, friends? Now let us see what happens in G1 phase. Let us write some important points that are occurring in G1, and we already know that what happens during S phase or synthesis phase, DNA replication occurs, right? So to make this DNA, we need enzymes, we need proteins, we need nucleotides, we need more hydrogens, we need many different substances. So all those substances which are required for S phase, they are synthesized here. Right? So what happens? First of all, nucleotide synthesis increases. Clear? During G1, inside the nucleus of this cell, what happens here, that nucleotide synthesis will increase. Why? Because we have to combine all these nucleotides to make an extra copy of the DNA. Right? So nucleotide synthesis increases during G1. Then to make these nucleotides and to make another DNA, some enzymes must be required. So what happens? Enzyme synthesis increases during G1 in the nucleus of the cell, right? Number 3, friends. Whenever enzyme synthesis increases, so these enzymes come from a protein, and that protein is synthesized by a special messenger RNA. So I think messenger RNA must be in synthesis, must be increased, right? So RNA synthesis increases. RNA synthesis increases inside the cell. More RNA is made, right? Then number 4, look carefully. Whenever all these things are happening inside the cell, so what do you think? Energy is less required or more required? So more required. So we say that ATP synthesis increases. ATP synthesis increases inside the cell. So whenever all these things are happening, all these functions require more energy. So there is more energy synthesis inside the nucleus and outside the nucleus in the cytoplasm, so that all these functions should be carried simultaneously, right? So ATP synthesis increases inside the cell. Clear, friends? So we said that during G1 phase, what happens that nucleotide synthesis increases because these nucleotides must be combined, and when these nucleotide synthesis increases, these nucleotides are sent to S phase. In the S phase, these nucleotides are combined and made to make another copy of the DNA. It's so simple. So for the synthesis of all these things, enzymes are required. So enzyme synthesis will increase and will jump to S phase another, right? And for these enzymes, we have to make a RNA. So RNA synthesis will increase, and so on. ATP synthesis will be increased. When all these things are happening, so the nucleus size will a little bit increase, or we say cell size increases. Cell size increases, right? So all these changes are occurring in G1, right? Done with it. Now let's come to S phase. What happens during S phase? All these things which are prepared in G1 are sent to S phase, and the enzymes are told to, we are sending different substances towards you. You please start replicating the DNA and make another copy of the DNA. So those enzymes will start working and will replicate the DNA. One thing. The next thing, what happens during S phase? The important thing, replication of the DNA. The next thing, what happens, that we have here a centriole. So this centriole is copied. Copy of centriole is made, and one centriole moves to one side of the cell, opposite one pool of the cell, and other centriole moves to other pool of the cell. So what happens during S phase? The first thing is DNA replication. The second thing is centriole copy is made and sent to opposite pools. Clear, friends? So these changes are occurring in during S phase. Whenever the S phase completes, the cell which came from the G1 was like this. Now whenever this cell is leaving the S phase, it's becoming like this, right? So what happened here? The cell which comes out of the S phase, this cell must have extra copy of the DNA. Its DNA must have been copied, replicated, right? So if we have here the DNA 2 capital N, right? So whenever the cell comes through the S phase, the DNA becomes 2 capital N plus 2 capital N, 4 capital N, right? So when the cell leaves the S phase, it must have 4 capital N DNA. The DNA must have been replicated. Clear, friends? Okay. So what happens during G2 phase? Nothing special, only the DNA is checked. If the DNA is okay, then this is sent towards the division phase. If the DNA is not okay, then some enzymes are activated to make the DNA, to correct the DNA. And if the DNA is not going to be corrected, then the cell is gone to apoptosis and the cell will die, right? So listen carefully. During G2 phase, what happens? DNA checkup occurs. When DNA is checked, DNA is okay, go towards the mitotic phase or division phase. Number 2, proteins are made, protein synthesis increases, right? Then some enzymes are synthesized, and whatever next things are required inside the cell, all those are synthesized. So enzyme synthesis increases, enzymes are formed in, and what happens? Cell size increases. So all these changes are occurring during interphase. Clear, friends? Now when the cell comes here, this cell which was here, it has single amount of DNA. When the cell comes towards the M phase, this cell must have double amount of DNA, double amount of cytoplasm, double amount of enzymes and other things, right? Cellular organelles. Clear? So look, here interphase finishes. So whenever interphase finishes, then the next phase of the cell starts, which is called the mitotic phase. So from here up to here, this whole phase represents the mitotic phase. Mitotic phase, right? So mitotic phase has how many steps? We will just write their names, but we will discuss these steps in our next lecture, right? So mitosis occurs in how many steps? The first step which occurs is called the prophase. Then the next step that occurs is called the pro-metaphase. And the third step that occurs is called the metaphase. And the fourth step is called the anaphase. And the last step that occurs is called the telophase. Clear? And the most important and the last step that occurs is called the cytokinesis. Clear, friends? Cytokinesis. So when these 4 steps occur, the cell is divided again. So what will happen when this cell will pass through these steps? Here will be the formation of how many cells? Here will be the formation of 2 cells. One copy of DNA is sent to one cell and other copy of DNA is sent towards other cell, and when cytokinesis occurs, the cell will lead to division and will make 2 other cells, right? So we should have a clear concept of this interphase, right? Next we are going to discuss about the mitotic pairs, that what happens during mitosis inside the cell and how the cell divides, right, friends? So see you in the next lecture regarding mitosis. Please, friends, if you like our lectures, please share these videos with your friends also. Thank you so much.